The impact of a warming ocean on aquatic life depends on the season, particularly for ectotherms that do not control their body temperature. Many of these creatures rely on environmental cues to regulate behavior such as metabolism. The timing of this process can be crucial for their survival. This study investigates the relationship between resting metabolism, temperature, and size using the brittle star as a model organism. Based on past research on marine ectotherms, it was hypothesized that brittle stars can withstand disproportionate increases in winter temperatures due to their ability to acclimate their metabolism. They predicted that acclimating wild sea stars to temperatures 3 degrees warmer than typical environmental temperatures would result in a 2 to 3 time increase in metabolism, a higher metabolic rate during arm regeneration, faster arm regeneration, that there would be a proportional increase in metabolic rate dependent on size at a higher temperature, and that it would not result in increased mortality.
In August, over 1,500 brittle stars were gathered at Little Bay, Sydney, Australia, during low tide with the average environmental temperature at 15.5 degrees Celsius. To assess increased metabolism at elevated temperatures, a treatment group underwent a gradual acclimation process to a temperature of 18.5 degrees, and that was compared to control data. To explore metabolism during arm regeneration, one group acclimated to 15.5 degrees and the other to 18.5 were subjected to arm automatization induced by forceps pressure. The resulting developing arms were measured at regular intervals, and development was assessed using photomicrographs. To investigate heightened metabolism in larger brittle stars at elevated temperatures, small specimens with intact arms were acclimated to 18.5 degrees and compared to larger brittle stars acclimated to the same temperature. Metabolic rate was assessed using closed chamber respirometry. This method involves placing brittle stars in sealed chambers and tracking changes in gas concentration. The reduction in oxygen level serves as the basis for calculating the rate of oxygen consumption. Data was analyzed using various statistical methods, including ANOVA and ANCOVA, to assess effects of size, temperature, and time on respiration rates and growth.
Our two significant findings showed an elevated temperature resulted in up to a 3-fold increase in respiration, and warm acclimation resulted in faster arm regeneration in terms of length and morphological features. Looking at the results of brittle star mortality, the brittle stars were acclimated to either 15.5 or 18.5 degrees and were categorized into whole and regenerating animals. Their survival rate was assessed over a period of 10 weeks. The trend indicates that whole animals have a higher survival rate than regenerating animals, and those acclimated to the environmental 15.5 degree temperature had a higher survival than those acclimated to the 18.5 degree treatment.
Although brittle stars demonstrate the ability to acclimate some of their physiological processes such as regeneration, the prolonged elevation of respiration rate and increased mortality during a simulated winter heat wave suggest potential threats to the species. This emphasizes the importance of understanding the impact of climate change beyond traditional seasonal expectation, and we suggest that this continues to be explored in brittle stars in a long-term study.